So the types of assays that cover this space are the traditional IVD assays, often being co-developed with the pharmaceutical and diagnostic company. They're often co-launched and co-approved and co-funded at the same time. So these are the classic HER2 immunohistochemistry, FISH assays, for instance, and the EGFR, RAS, and BRAF mutations. They're usually single gene assays, and they're very precise and very specific. Then there are next generation sequencing based small targeted panels that may have anywhere from a few to a few dozen. So these can tile different parts of the cancer mutation landscape. So what we're seeing today is laboratories using this next generation sequencing technology to amalgamate these single IVD assays into single panels. So a panel can be designed that may contain EGFR, RAS, and BRAF, for instance. And so these types of small panels, which are more economic and more information is gathered, are becoming increasingly used in clinical practice. They often cover all clinically relevant or actionable biomarkers relevant for common tumor types, but they may contain other genes of less or uncertain clinical significance. If genes which are responsible for inherited cancer syndromes are included in the panels, then you may uncover unsuspected germline mutations such as BRCA1 or BCR2. Now, because some of these small panels are often replacing the IVD assays, they need to work on all specimen types. The size of the specimen, the amount of DNA, the tumor content, and then different qualities of specimens. They need to be validated on the full range of specimen types which are likely to be encountered in clinical practice. And because these are being used in the precision medicine space, and we want to avoid false negatives, it's essential that we get the right result first time. And because they're replacing already reimbursed tests and they will be funded accordingly, they need to be cost effective and not too expensive. And because, again, these are being used in the precision medicine space as replacements or surrogates for the IVD assays, we need to be able to have the results quickly so that they're ready for the NDT meetings.